How?
PARP-1 is an abundant nuclear protein critically involved in numerous biological processes linked to cellular stress responses. The enzymatic function of PARP-1 is to catalyze a protein post-translational modification called poly-ADP-ribosylation, PARylation. PARylation consists of linear and/or branched repeats of ADP-ribose, with lengths reaching up to 200 units. Compared to other common PTMs, a unique feature of ADP-ribosylation is that this modification can occur on amino acids with diverse side chain chemistries, including ASP, GLU, LIS, ARG, CIS, SER, and TER. Besides PARP-1, numerous efforts have now identified 16 additional PARP enzymes. PARP-1 and PARP-1-mediated PARylation events are tightly connected to the DNA damage response, DDR. The PARylation level in a quiescent cell is usually very low. In response to genotoxic stress, PARP-1 binds to nicked DNA and is rapidly activated. This leads to the generation of a large number of PAR-related proteins and, subsequently, the initiation of the DNA damage repair program. From a structural perspective, PAR resembles DNA, both being bulky, charged, and flexible. The interaction between DNA and a DNA-binding protein could therefore be disrupted due to its covalent modification by PAR. In addition, PAR polymers may also act as a scaffold for recruiting other proteins that bear PAR-binding motifs, PBMs, including OB, WWE, PBZ, BRCT, and macrodomain. These PBMs bind to different structural units within PAR, and they are present in many proteins involved in DDR. The critical role of PARP-1 in mediating DDR provides the rationale for developing PARP-1 inhibitors to treat human cancer. BRCA1/2 are tumor suppressor proteins that play a critical role in mediating DNA double-strand break, DSB, repair. They are mutated in about 10% of breast cancers and 15% of ovarian cancers. It has been shown that BRCA1/2 mutated cancer cells are deficient in homologous recombination. As a result, they rely on PARP-1 for genome integrity and can be selectively targeted by PARP-1 inhibitors through the concept of synthetic lethality. Besides blocking the formation of PAR polymers, recent studies suggest that PARP-1 inhibitors, PARP-I, may also kill tumor cells via a trapping mechanism. Upon sensing nicked DNA, PARP-1 is activated and automodified. Auto-PARylation of PARP-1 results in its dissociation from DNA due to steric hindrance and charge repulsion. When treated with PARP-I, PARP-1 cannot modify itself and is trapped at the DNA lesion. This PARP-1-PARP-I DNA complex interferes with subsequent DNA replication, making it highly cytotoxic. Although this model suggests that catalytic inhibition of PARP-1 is functionally inequivalent to PARP-1 deletion, there lacks a pharmacological approach to dissecting these two pathways, i.e., PARP-1 catalytic inhibition versus PARP-1 trapping, to assess their relative contribution to PARP-I cytotoxicity. Besides its role as an oncology target, PARP-1 is hyperactivated upon sensing genotoxic stresses associated with various pathological conditions, such as ischemia-reperfusion, IR, injury, and neurodegeneration. Overstimulation of PARP-1 could account for more than 90% of the PAR synthesized under these conditions, representing the primary mechanism for NAD+ catabolism. Depletion of the cellular NAD+ pool forces NAD+ synthesis through the salvage pathway, lowering the cellular ATP level and eventually causing necrosis. Recent studies have shown that the accumulation of PAR could also lead to the translocation of apoptosis-inducing factor, AIF, from mitochondria to the nucleus, causing another form of cell death known as parthenosis. PARP-1 is thus also being evaluated as a therapeutic target for IR injury. However, due to the significant cytotoxicity associated with PARP-1 trapping, PARP-1 inhibition with current inhibitors may not be useful in this context. The development of selective PARP-1 inhibitors that mimic PARP-1 genetic depletion, i.e., inhibit PARP-1 without causing PARP-1 trapping, will likely have substantial therapeutic potential. Recently, several groups have reported a strategy in which a small molecule ligand of a target protein is linked to a binder of a ubiquitin E3 ligase. This bivalent agent brings the target protein and the E3 ligase into proximity, leading to protein ubiquitination and subsequent degradation through the proteasome pathway. This strategy, known as proteolysis targeting chimera, PROTAC, has been designed using small molecule binders of various E3 ligases, such as cereblin/CRBN, VHL, and MDM2. By conjugating these binders to a ligand for a protein of interest, induced protein degradation has been demonstrated for numerous targets, including RIPK2, BCR-ABL, BRD4, and CDK9. PROTACs offer a range of unique advantages over catalytic inhibition, including greater versatility, a plug-and-play mode of action, prolonged effect, and the ability to inhibit the activity of undruggable proteins. Importantly, by pharmacological depletion of a protein of interest, PROTACs can abrogate the non-enzyme-dependent, e.g., scaffolding, function of the protein. #### PARP proteins. ADP-ribose, polymerase 1, PARP-1, also known as NAD plus ADP-ribosyl transferase 1 or poly-ADP-ribose, synthase 1, is an enzyme encoded by the PARP-1 gene in humans. It is part of the PARP family of enzymes. PARP-1 modifies nuclear proteins through poly-ADP-ribosylation and works with BRCA in double-strand DNA repair. Members of the PARP family also act on single-strand breaks or, in cases where BRCA fails, PARP takes over. PARP-1 is involved in differentiation, proliferation, tumor transformation, and DNA damage recovery, both normal and abnormal. It also plays a role in the pathophysiology of type I diabetes and may be linked to mutations in Fanconi anemia. PARP-1 is activated by Helicobacter pylori in the development and proliferation of gastric cancer. PARP-1 is crucial in repairing single-stranded DNA, ssDNA, breaks. Reducing PARP-1 levels with siRNA or inhibiting its activity with small molecules decreases ssDNA break repair. Without PARP-1, when these breaks occur during DNA replication, the replication fork stalls, leading to double-stranded DNA, dsDNA, breaks. These breaks are repaired via homologous recombination, HR, a potentially error-free process. Cells lacking PARP-1 exhibit a hyperrecombinogenic phenotype, showing an increased frequency of HR, as observed in vivo in mice. However, if HR is functional, PARP-1 null mutants do not display an unhealthy phenotype, and PARP-1 knockout mice show no adverse effects or increased tumor formation. PARP-1 is one of 6 enzymes required for the error-prone DNA repair pathway, microhomology-mediated end joining, MMEJ. MMEJ is associated with chromosome abnormalities such as deletions, translocations, inversions, and other complex rearrangements. PARP-1 upregulation increases MMEJ, leading to genome instability, which is observed in tyrosine kinase-activated leukemias. PARP-1 is overexpressed in several cancers when its promoter region ETS site is epigenetically hypomethylated. This contributes to endometrial cancer progression, BRCA-mutated ovarian cancer, and BRCA-mutated serous ovarian cancer. PARP-1 is also overexpressed in cancers like neuroblastoma, HPV-infected oropharyngeal carcinoma, testicular and other germ cell tumors, Ewing's sarcoma, malignant lymphoma, breast cancer, and colon cancer. PARP inhibitors. PARP inhibitors are a class of drugs that inhibit the enzyme poly-ADP-ribose polymerase, PARP, and are developed primarily for cancer treatment. Many cancers rely on PARP more than normal cells, making PARP a valuable target for cancer therapy. PARP inhibitors have shown potential in improving progression-free survival in women with recurrent platinum-sensitive ovarian cancer, particularly when combined with treatments like olaparib. In addition to cancer therapy, PARP inhibitors may be useful in treating acute life-threatening conditions, such as stroke and myocardial infarction, and chronic neurodegenerative diseases. DNA undergoes thousands of damage events during each cell cycle, requiring repair, including in cancer cells. BRCA1, BRCA2, and PALB2 are crucial proteins in the error-free homologous recombinational repair, HRR, of double-strand DNA breaks. Mutations in these genes can lead to DNA repair errors and eventually cause breast cancer. Significant DNA damage can cause the death of cells with mutated BRCA genes. PARP-1 is essential for repairing single-strand breaks. If such breaks remain unrepaired by the time DNA replicates, replication can cause double-strand breaks. PARP-1 inhibitors lead to the accumulation of these breaks, and in tumors with BRCA1, BRCA2, or PALB2 mutations, the resulting double-strand breaks cannot be efficiently repaired, leading to cell death. Normal cells that do not replicate as rapidly and lack BRCA mutations can still use homologous repair to survive PARP-1 inhibition. PARP-1 inhibitors trap PARP proteins on DNA, in addition to blocking their catalytic function, which interferes with replication and causes cell death, particularly in fast-growing cancer cells. Some cancer cells lacking the tumor suppressor PTEN may be sensitive to PARP inhibitors due to RAD51 downregulation, although there is conflicting data on whether PTEN regulates RAD51. PARP inhibitors may also be effective against PTEN-defective tumors, such as certain aggressive prostate cancers. Cancer cells with low oxygen levels, as found in fast-growing tumors, are also sensitive to PARP inhibitors. Currently approved PARP-1 inhibitors include olaparib, rucaparib, niraparib, and talazoparib. Other inhibitors in clinical trials include veliparib, CEP9722, E7016, BMN673, and BGB290.
PharmaPal.